Good morning, everyone. Hi. Thank you for joining me on this Friday morning. The topic of today's discussion is actually quite an interesting topic. It's not a didactic lecture around a particular internal medicine a clinical entity or disease syndrome, but rather it's what the Department of Internal Medicine has to offer our colleagues on the platform. So we've been working quite hard over the last, 1.5 years to try and develop a standalone research unit within the Department of Internal Medicine. We, unfortunately, unlike what the research units have overseas, we don't have a standalone research unit that has a sort of a conveyor belt of research. And what we've been trying to conceptualize is this sort of a research structure. And around this particular, vision, we've, with the research professors that we have in our department, and we've been blessed to be given 2 new ones, is we thought of developing a division within the Department of Internal Medicine as a standalone division. So why is that important for the clinical platform? It's important to the clinical platform for several reasons. One is that it offers us a focus for dedicated research. So the staff that are working in this particular unit are no longer support staff. They actually are going to be embarking on research projects in which anyone is willing or anyone who has a research interest should participate in. Furthermore, our registrars, our medical officers, and even our students have all been looking for research projects to improve their CVs, to improve their academic outputs, and most importantly for their degree purposes, right? Very soon the MPhil is going to be, mandated by the college. And as we know, a research dissertation is mandated by the college for the FCP. Doing retrospective research in the Department of Internal Medicine is always challenging because we don't have an electronic data, electronic database. So it creates quite a bit of a problem for students to mine, data that's actually verifiable and has robust information in it. So within this research unit, we are now trying to create databases, use existing databases that have, data that's been cleaned and that's ready for interpretation or for analysis. So without further ado, I'm going to introduce you to the research professors. Prof. Mahdi has been kind to internal medicine, and we should actually use this opportunity because I don't know how long we're going to be actually supported in this particular capacity. But we've had 2 new research professors that have been appointed, Professor Alisha Wade, Professor Zané Lombard, and I'm going to leave it to them, each of the colleagues on the particular platform whose names are up on the slide, to present what their specific research interests are and to try and spark a curiosity in the audience around something that you think you might want to pursue as a research topic. And then feel free to approach them, create or conceptualize a paper, conceptualize an MEd dissertation, and even work towards a thesis. So, it's become much more, it's no longer a pie in the sky getting a PhD. The school has gone through extensive lengths to try and make a PhD much more accessible to the students on the platform, even intercalated degrees. So I think with time, I know Vida has already got 2 students that are within their medical careers that have taken 2 years off and are now going to qualify with 2 degrees. And if we can get this, research entity as a standalone, conveyor belt of research, I'm sure we can plug in students to complete a dual degree, so get an MBBCH and a PhD for me. So without further ado, I'd like to introduce our first colleague, Alice. She's from the Hepatitis B Viral Diversity Unit, and she's looking to repurpose herself outside of Hepatitis B research and to offer her support to the clinical platform. Alice, over to you.
So from my career to now, my interest is quite wild. I started with virology, in virology, so I did quite a lot on hepatitis viruses, the HIV viruses, the Epstein-Barr viruses, the co-infection, and the super infection of viruses. We also looked at the viral and host interactions, like viral interference, viral synergy, and viral non-interference. What we also did is I did a lot of genotyping and molecular characterization of viral strains. We also look at, before we look at the viral and host response to antiviral or anti-tumor therapy. Then later on, I moved to look into pathogen evolutions and biology, looking at bioinformatics, using it to identify outbreaks, identify the origin of pathogen, and also to track the spread of diseases. For example, we did one study to look at the mesocomal infection in dialysis unit. And then I also, besides on the virology, and then I moved into look at gene expression, gene mutational analysis, and also in my PhD, I did a lot on tracking on intracellular protein processing and movement. Okay, so the laboratory techniques we have in the molecular side is very molecular intensive. It's molecular cloning. We construct expression vector for in vitro expression in culture cell lines. Mostly, the other experiment techniques we use a lot is the endpoint PCR and real-time quantitative PCR. We also do sequencing, the Sanger sequencing for usually for small scale project or confirmation of the next generation sequencing results. Protein analysis, protein expression, extraction and purification of bacteria and culture protein from bacterial or culture mammalian cells, and also detection of protein immunoprecipitation, protein electrophoresis, Western blot, ELISA. In addition to that, we are able to do the immunofluorescence and confocal fluorescence imaging, and then lastly, the in vitro cell culture. So at the moment, I have 3 projects that I try to branch out from HBV. First is to develop a custom nephrotic syndrome panel for sub-Saharan Africa populations. And the second one is looking at the EBV infection in nephrotic syndrome patients. And my last project at the moment is also looking at the EBV-related human malignancies in mono-infected or co-infected individuals. So this will be like a more retrospective study, try to look into the database and see any change over time, the prevalences, and what doctor will order in each unit, what type of test do they order. So that's my research interest at the moment.
Thank you very much for that, Alice. So just again, so I forgot to mention that this particular unit is available to the entire clinical medicine platform, right? So it spreads across our entire platform, and including our satellite units in our extended academic platform. So Tsepong, Clarkstop, Sebokeng, Berthagoa, Edenvale, wherever you might be, if you have a research interest and you're looking for someone to assist you in developing that particular project, please feel free to come through and speak to anyone in the research division. And they'll be able to find someone. We've got someone in every aspect of research at the moment will be able to facilitate some sort of research project, right? So anything that ends with the omics, so genomics, transcriptomics, proteomics, we can facilitate any research in that particular capacity. Right? Thank you very much for that, Alice. So basically, Alice's field of expertise is around molecular subtyping and PCR technology. If anyone has some ideas around that and is looking for any particular or has a research topic around not necessarily just infectious diseases, but particular infectious diseases, please feel free to come and speak to Alice. Alternatively, if there is a bit of a concern about how to approach the research unit, I mean, you're more than welcome to liaise through my office, through Marlies, through Busi, and then through the research professors if they might be away on a research project. So please don't let that deter you from wanting to drive your research agenda. Okay, so the next one is Raquel.
Hi, my name is Raquel Duarte, and I thank you, Alice, for giving a very, very good overview of the sort of techniques employed in the molecular biology research lab. So I just want to mention that I'm part of an amazing group, which Therese, Dr. Dick Speak is here, and Dr. Dickens, and then we have our technicians, Confidence and Donald. So as we mentioned, we've been responsible for the molecular-based research up to now. However, we're branching out and concentrating all our efforts in building the translational research unit for the medicine. So why do we consider translational research? I thought this was the best umbrella in which we can embed all the activities that we're involved in. And traditionally, we know translational research is a sort of bench to bedside model. However, we've taken on this model proposed by the NIH group NCATS, which shows that it's more complex than initially suggested, and that translational research can take place in any one of these nodes, and as long as it's in the patient treatment continuum. And the important thing is that there's information constantly flowing to and fro. So what I'm going to do now is just show you some of the research we're doing in these particular nodes, and hopefully you can get a feel for the type of work we're doing. So the first, of course, is the bench one, bench basic research. And this is my particular interest, and that's in understanding triple-negative breast cancer. To this end, I've screened a number of triple-negative breast cell lines and identified those proteins that are differentially expressed. Identified one, which is known as KSRP. Very interested in it because it's an RNA binding protein, and very interested in how it impacts alternative splicing. And this here is a diagram of pre-messenger RNA, which is one of the top hits in breast GWAS. And this is the sort of mechanism that underlines alternative splicing of exons that will eventually make up your protein, the work end of the molecule. And this just shows you that the various factors that are involved in the splicing and silencing and underplays the expression of this protein and sets up different subtypes. So we want to see how our protein, which is indicated in orange, we've shown that it first of all binds it and overlaps that little silver protein, a silencer PTB. So we're trying to unravel this complex mechanism and see how we can switch subtype, obviously for therapeutic function. The second one is the bone induction. This is part of tissue regeneration, where we've helped understand the molecular mechanisms underpinning a bone induction model, a spontaneous one. And then you can see this beautiful, beautiful blue lamellar bone that's formed in these concavities of calcium phosphate bioreactor. And we've been responsible for the gene expression profiling of a number of the pathways and subtypes that make up this model. Dr. Asaid Al-Khanti is now looking at cell types of the environment. And the whole reason for this is to create a second generation bioreactor that can then be used to treat submandibular defects. This has gone into a preclinical model, so you can see that flow of information. The second lot of work, which goes place from the clinic to the bench side, is the work that's been carried out by Therese, where she's looked at molecular subtyping of breast tumors in our population. The way she's done that is with the PAM50 assay. It's a genomic assay, which subclassifies tumors using a signature of 50 genes. So based on that differential expression of the genes, you're able to classify tumors into one of those subtypes. She's described the subtypes in our population, and has used it also to inform and look at the current strategies for immunohistochemical diagnosis of breast cancer in South Africa. So our efforts are now continuing to look and characterize more the major subtypes in South Africa that the HER2 and the BAs are like. The next project, I'd like to show how we develop projects and work with our clinical partners. And this is the work of Dr. Muzamil Hassan. He's looking at kidney dysfunction in HIV-positive adults. He screened a cohort of 3,000 patients to see who those developed nephrotoxicity as a result of tenofovir use. We're his lab partners, and we've developed these projects where we've done extensive biomarker analysis. And because it could, the way they metabolize the drug could be causes the toxicity, we've carried out a pharmacogenetics component. We've done SNP genotyping, looking at one of the main targets of kidney dysfunction. Now, the whole reason for all this research is to build a signature similar to what you've seen with the PAM50, but it's able to predict which patients are going to suffer from nephrotoxicity. We're very interested now going forward in using the cell-free components, and that's your serum and urine, especially for diagnosis and using prediction. So we've carried out a genome-wide next-gen sequencing profiling of microRNAs and identified 3 microRNAs that could distinguish between those that develop nephrotoxicity and those that don't. And we're now validating this in a larger cohort. So the last sort of big study that we're involved in is looking at nephrotic syndrome. And this shows you a beautifully differentiated podocyte, which is responsible for maintaining the membrane activity of your glomerulus. And we're part of an international study, which is the Nephros group, which is led by our colleagues in Bristol. And we've collected an extensive database as well as clinical biospecimen sampling. This is a project that's been initiated by Graham Paget, where he's looking to see the cause of idiopathic nephrotic syndrome, and is looking at if it's possibly due to a maladaptive response to EBV infection. What it basically means is that antibodies developed during the course of an EBV infection somehow targets any of those proteins that are involved in maintaining the skeletal structure of this podocyte. And what we've done is we've looked at we've carried out EBV serology, we've looked at EBV viral levels, and because it could have an immune genetic background, we've done extensive HLA typing. We will now follow this on with a bioinformatics component where we'll look at the relationship between the EBV proteins and those of the kidney, which will then inform our lab-based work where we'll look at cross-reactivity of EBV proteins with some of those skeletal proteins. Aside from this, you've already mentioned some of the work that Alice is talking about, where we're also doing exome sequencing of this is with the colleagues in Bristol, and the data of which will be used to determine what variants or mutations are present in our population, all for the benefit of developing the next-gen gene panel used for diagnosing the prominent forms of nephrotic syndrome in our population. So I hope that gives you a bit of a taste of the sort of projects we've been involved in and the sort of clinical partnerships we have. And I'd just like to thank you, Andrew, for that quote.
Thank you very much, Raquel. Are there any questions for Raquel or for Alice from the panel or from the audience? We can, yeah, we'll take care.
Okay, all right.
Okay, all right. So just to build on what Raquel said, I don't know if any of my GEM4 students are on the platform who I did a talk with yesterday, or if they are. So we saw a very interesting patient yesterday. She was a 31-year-old female who had breast cancer. Most said, diagnosed in November last year, is now got metastatic stage 4 breast cancer at the age of 31, with no significant risk factors, RBD negative, you know. And that's just a case in point. Now, those particular students, Raquel has just presented a very nice analysis of what she's doing with breast cancer. And if this sort of a clinical patient that you saw clinical scenario sparks a field of interest in you, and you want to learn a bit more about it, then you're welcome to come and discuss this particular aspect. And even if you want to try and identify, molecular signature around why this particular patient is behaving in this way, this is where we're doing all of this work. This is the back end of the work that's leading to these particular manifestations that we've seen on the clinical platform. What we need to understand, and just for me to interject, is that medicine is becoming more individualized, but within that individualization of medicine, there are different molecular signatures that we need to try and identify. And it's only us on the clinical platform that identify or can see these signatures. So, keep an open mind to it. If you pick up something anywhere, any small, and it sparks a seed of inquisitivity, please, try and explore it and come back to us and see how we can assist you in developing a research project around it. So I'd like to introduce Zané Lombard. She's just been appointed into our department as a scientific clinical research professor, and she's from the Department of Human Genetics. Over to you, Zané.
Thanks so much. And I'm really glad that Raquel went before me and that Prof has given this introduction, because it speaks so well to the type of research that I'd like to initiate and build on here in internal medicine, now that this is my new home. And I think we've kind of heard the beginnings of this, but this is what I see as kind of the benefits of genomics and what it can bring to healthcare, and specifically here in internal medicine. I think this is really a really good base to start building this kind of genomic research that would benefit not only the individual patient, but it can also have a benefit at the population level. So I think genomics has really grown as a field from not just being a very niche specialized area where it's very focused on maybe a smaller niche specialization, but a field where we can use genomic information to help patients to do better diagnostics, to do earlier, faster, and more accurate diagnostics, but also to use that information then in a prognostic way and to optimize treatment, use individual information on the genomic level to optimize outcomes, to make sure that we're targeting drugs in a way that we have less adverse drug effects in our patients. And then I guess at a population level to really start using this information in a preventative way. So to try and identify groups of individuals that are particularly high at risk for certain diverse disease outcomes, and also to use that information ultimately not only to intervene and to start early treatment, but ultimately also to try and prevent some of these complex diseases where we have both the genetic as well as an environmental component. So why would we want to do this here in Africa? And I think there's a really important and exciting gap in our knowledge at the moment when we look at the kind of genomic research that's happening across the globe, in that the majority of it, more than 80% of it, is very European ancestry focused. The reason why this is creating a gap is not only is this inequity hampering us from implementing genomic medicine for local African populations and for other populations outside of the European ancestry cohorts, it's also speaking to genomic diversity. We know that Africa is seen as the cradle of modern humans, and that the kind of genomic diversity that we can explore and see in African cohorts is significantly expanded from what we know is in some of these individuals who are living outside of the African continent. The reason why this is important is because if we think about everything that we know up until now in genomics that is applicable to medicine, from studying a majority, a subset of the genomic diversity, if we shift our focus to African cohorts and exploring additional diversity in that sense, there's so much more that we can learn. And this could really not only expand our understanding of disease for local populations, but also have a global impact. Okay, so I think we can definitely see that genomics can be implemented locally. I think the COVID pandemic has given us a really good example showing how African researchers can lead this kind of research, where researchers from South Africa, from Nigeria were at the forefront of doing genomic surveillance and sequencing, training individuals, moving forward with sample sharing, with biobanking. And this is a prime example to show that the kind of infrastructure that we need to do this kind of research is already in place. And we can just utilize this to move forward and do some really interesting and important research here in internal medicine. So some of the gaps that remain and some of the work that we can be doing together here is to really start focusing on things like common disease genomics, where access to diverse and well-defined cohorts is really important. And these are the patients that you see each and every day. Some of the interesting cases that come up, there is interesting genetics questions that can be asked. And this is a great opportunity for building partnerships and building those cohorts that can be used for additional genomic research. With rare disease genomics, again, the expertise is in place, but I think we need to expand that, train additional people, build on the expertise and the knowledge that we have by training and then developing additional posts. And I think something that is really key here and something that I'm really interested in building up is the data analysis tools and the data analysis expertise that we need to analyze this data. So genomics has become very data science focused. These are all big data sets. And I believe building out our expertise in clinical bioinformatics, and that is where we learn to use computational tools to answer specific clinical questions is a niche area that we can really excel at here at the department. So my final slide, I just wanted to introduce you to an existing study that I'm currently running that is called Deciphering Developmental Disorders in Africa, or DDD Africa. This is a specific study looking at the genetic etiology of developmental disorders in a pediatric cohort from South Africa as well as the DRC. We're collaborating with individuals from the Wellcome Sanger Institute in the UK and KU Leuven in Belgium. And with this particular study, we've built up resources both from a clinical, a laboratory, as well as a bioinformatics perspective, putting things in place that can be extrapolated and easily used in additional studies. But there's also now this existing resource that can be accessed for specific MH projects, etc. So I'm happy to speak to anybody who might be interested in looking at this data and thinking about some of the questions that can be used for new research projects. And also just to mention, this is an ongoing project. So again, if this is something that piques your interest, I'm happy to speak to anybody who'd want to join the project and continue with that. Thank you so much.
Thank you very much for that overview, Zané. So just to explain a little bit around what Zané explained, we're all familiar with the Mendelian inheritance of disease entities, right? Where there's a clear familial, but what we don't understand is that there's a significant genetic component to diseases that might not necessarily be evident to us, but we can just pick it up because there's a significant feel for it. We understand, that there might be some clustering within a particular specific population group. And just that particular seed of inquisitivity again can lead to us developing, a molecular signal that allow us to individualize the genome, that speaks to that particular disease entity, and then from that develop individualized therapies or individualized diagnostics, which speaks to the previous two speakers. So I think it's exciting work, and we need to we need to be at the forefront of this. This is what's happening everywhere else in the world. We're actually falling behind in Africa. And if we don't embrace this technology and we don't keep up to times, we're still going to lag behind in terms of our conceptualization of disease entities that are peculiar and unique to our environment. Okay, so next up on the list is Aadilah. Okay.
Good morning, everyone. I'm Dr. Omar. I am an emerging researcher in the field. So this is just very preliminary stuff. What I'm looking at the moment is immune evasion in colorectal cancer stem cells. This project is currently in progress. I've got an honors student working on that at the moment. What I will be going into using NRF-Tutuka funding is looking at the sonic hedgehog signaling in colorectal cancer and crosstalk with the BRAF pathway. So this project will be starting, next step will be a 2-year project. I'm looking at recruiting some students for that and collaboration with University of Pretoria as well as UJ. So the immune evasion in colorectal cancer stem cells project is using commercially available established cell lines. So, the relevance of this, it's interesting to see and to explore this, but I want to see what's happening locally with South African patients. So this project here will actually be looking at getting local patient samples and growing these or establishing these cell lines and working on these. The benefits of this, as Zané mentioned, exploring it, using patient samples derived locally, it puts it more, I find it more relevant than using these commercially available cell lines. And in terms of diagnosis, therapeutics that can be developed for that and its relevance in our context within South Africa, I think is important. Moving forward, I'll be looking at exploring the interactions between lung cancer, HIV, and TB in South Africa. So this will be from 2027 onwards. I'm still in the design process of the study and sourcing of funding. But if there's anything of interest to you guys in terms of participating in these projects or helping in the development of these projects, please come and reach out to me and have a chat. I am looking for some inmates to help with a few things as well. So yeah, please reach out to me. In terms of techniques that are currently being used and taught, so a lot of cell-based assays, cell culture. If you need assistance with training in cell culture, I can assist with that. So yeah, a lot of cell culture-based assays looking at cell viability, proliferation, signaling, gene expression, etc. The cancer stem cell project utilizes magnetic cell separation in order to extract these stem cells and work on them. Real-time qPCR, ELISAs, as well as fluorescent microscopy is all part of these projects. And then looking forward, the techniques that I would like to incorporate, so there is a bit of NGS happening, but I do want to continue working on that. But sort of introducing or developing CRISPR-Cas9 in terms of a gene editing tool. And there's something that really sparked my interest that I wish to develop in the department and explore in collaboration with UJ is looking at using machine learning. So you look at compounds, take data or information from literature, lab research and patients, and you obviously use AI and machine learning to develop therapeutics and look at that. So if there's anything in that you guys are interested in, please reach out to me and have a chat. Thanks.
Thank you very much, Aadilah. So just to conceptualize what Aadilah is speaking about with regards to CRISPR, I don't know if anyone has read the NEJM articles about the hemophiliacs. So basically, we're looking at, patients who have extremely refractory therapeutic options at the moment. So they develop complete resistance to the current therapies that we have for them. They've been hemophiliacs their entire lives, and with the recurrent transfusions that they've been getting, they develop antibodies against them. And now with this new gene splicing technology, most of these patients actually go into complete remission, actually become cured of their illnesses. This project's also been, rolled out into familial hypercholesterolemia. So there's a significant impact on clinical lives on our patients, and it's phenomenal. You can basically look at patients who've been taking infusions their whole lives, even our thalassemics, our sickle cell diseases. I mean, that's where this emerging technology is going towards, and it's basically looking at rearranging the gene abnormalities that cause the primary disease entity, and actually hoping for cure in these patients. So very conceptual, very relevant and current 21st century medicine that's been practiced, and possibly that's what we're looking at trying to introduce on our platform as well. With regards to oncology, so just to enlighten the audience, I think the two major focuses, other than in terms of communicable and non-communicable diseases going forward into the 21st century, would definitely be oncology, and then infectious diseases, right? And specifically infectious diseases, viral illnesses, which have been neglected, right? And in oncology, it's said that within the next 10 years, 1 in 4 people in the world will be diagnosed with some sort of an oncological problem. It doesn't matter what it is, whether it's something simple, something benign, or more malignant, right? But they'll have a clonal proliferation of cells. And that's quite a frightening statistic. It's going to outnumber everything else that we've come to today. And currently, our therapeutic interventions for oncology is limited. At best, we're curing about 15% of our patients and putting the rest of them in remission. And our toxicity from most of these therapies are quite significant. So I think it's not just an emerging, it's a well-established division in internal medicine, often neglected. And the burden of disease, I mean, if you guys have to go up to the oncology clinics, I think they're by far the busiest clinic that we have, because oncology is actually meant to be subdivided into each of the organ systems and the malignancies that are evident in that. Yet it's practiced under one division, and it spans the entire human body, right? So for people who have a significant focus in oncology, again, this is a significant amount of work that we're doing. There's the Duarte unit that's been developed at WITS. So there's a lot of work that's been done. Please, just to keep your eyes open, keep your ears open, and for any of you that are interested, whether it's at a molecular level, whether it's a clinical level, and you want to take back the work to a benchside, that it's happening. So please just remember that, right? The next is Clem. Clem also has got a significant oncology focus. Yeah, sure. Okay, all right.
Thanks very much.
Thank you.
Right, so morning everyone, and thanks for this opportunity to tell you about our work in the oncology research area. So I represent or head the Molecular and Cellular Oncology Research Group. The research as such is driven by an active postgraduate team of students, as well as a number of postdoctoral fellows, one of whom is Aadilah, who has contributed quite a lot in terms of the sonic hedgehog signaling pathway. So we have both a basic science approach as well as a patient-centered approach. And we are particularly interested in molecular and cellular aspects of cancer research. So a key focus are signaling pathways, crosstalk between the tumor and microenvironment, and now there's a new catchphrase, the tumor-immune microenvironment. Tumor initiation and progression, metastasis, the immune system, and of course PD-L1, which is a very hot topic for therapy at the moment. Just briefly in terms of what our key research focuses are in terms of the patients, it's been on early onset colorectal cancer in South African patients. And we have noticed, or we've actually recorded in several studies that Black South African patients are diagnosed with colorectal cancer about 10 years earlier than any other South African counterpart. So we're very interested in why this is happening. We've done a few genetic-based studies. We have also had a liquid where we're following the evolution of mutations over time. So that's a focused next-generation sequencing approach. And in addition to that, we are also profiling the microRNAs. Does the microRNA signature change over time? So when the patients are first seen, 6 months, and then that's 12 months down the line. Now we have a new focus in which Prof. Kalla mentioned, this crossover between viral infection and oncology. And I was fortunate enough to obtain funding from Aadilah to investigate the influence of the microbiome in early onset colorectal cancer in Black South African patients. By microbiome is an inclusive term of all microorganisms, including viruses and bacteria. At this stage, our focus will be on virus infections or co-infections in colorectal cancer patients. And why are we considering viruses? Well, if you think of sub-Saharan Africa and HIV infection, there's not that we're not particularly focused on HIV, but patients who have HIV infection are 2,000 to 3,000 times more likely to develop a cancer, to progress towards a carcinogenesis. And there are a couple of viruses that in recent years have been reported to be associated with colorectal cancer. And this includes Epstein-Barr, CMV, John Cunningham virus, among others. So we're at the beginning stages of this project. I think it's quite an exciting project. It's going to involve a focused next generation sequencing approach. We'll screen patient samples. We will assess whether or not there are viral infections, or at this point in time, we don't know whether these viruses might be passengers, simply just there because the immune system is depressed, or if they are actually playing a role in this early onset colorectal cancer. Now, just to briefly discuss a bench-based project with you, because it describes some of the techniques that we've used in trying to understand what we call the secretome. So these are secreted proteins. These are proteins that are secreted from cancer cells into the circulatory system. And why are they important? These are important because they influence the tumor stroma, the surrounding microenvironment of the tumor cells. And they're also able to influence, these proteins are also able to influence the immune system, signaling within the immune system and impede the immune response. So as I mentioned, this is a cell-based approach. We isolated a particular population of stem cells using magnetic cell separation. We collaborated with our partners at the CSIR, who analyzed our protein extracts using mass spectroscopy. And we were able to obtain a protein signature and to identify the proteins involved, the different populations or different groups of proteins secreted from the stem cell population versus the non-stem cell population of the cancer cells. And so we've been able to quantify the proteins, carry out some proteomics, gene ontology and pathway analyses. And at this point in time, we now need to validate the proteins. So we need to say, or we need to confirm that yes, indeed, these proteins are being expressed in these different cell populations. And so now I'm going to talk to you or tell you how we do this validation and how this might, how this might relate or help researchers within our department. So we principally use laser scanning confocal microscopy with a component called super resolution. And what this microscope does, it uses lasers to optically slice your samples and then software to reintegrate those slices into a 2D or 3D image. And this reconstruction allows us to very specifically analyze these cells at a single cell level. Now, just to mention that the microscope is housed in an existing unit within the faculty. It is managed by Dr. Orly Deribay. I've been the PI on the microscope and have, for many years, has directed that the unit and sort of have been involved with ensuring that the microscope actually stayed on at our faculty. We also do collaborate with the Faculty of Science, the counterpart in the School of Molecular and Cell Biology, and that is Professor Mandeep Kaur. So this is a collaborative instrument, if you like, used by science and medicine, as well as many outside users. And we would love to attract people from internal medicine to come and use this instrument to answer their clinical research questions. So how do we do this? How do we visualize these proteins? It's a technique called indirect immunofluorescence, where we use a primary antibody specific to our protein of interest. And then we detect our primary antibody using secondary labeled fluorescent antibodies. And this technique is very advantageous compared to a conventional microscopy technique because you can carry out multiple antibody labeling and detection on a single section or on a single series of cells. So here you can see the nucleus, there in red, cytoskeleton, and the cell membrane in blue. So it's not just a microscope. People say, when you talk about the microscope, people, oh, it's just microscopy. It's more than just microscopy. This is an analysis tool. This is a serious analysis tool which allows for biomarker validation. You can quantify your target proteins. You can look at pharmacological responses and responses of your cells to gene knockdown. You can interrogate the response of patients' tissues to therapies. We can look at changes in biomarker distribution. So, for example, some of these pathways, some of these transcription factors will relocate. So after drug treatment, they'll change their distribution from the cytoplasm to the nucleus or vice versa. So it gives you a lot of clues about how these pathways function, how your therapies are working or not working. Also, we can tell a lot about protein-protein interactions. So, for example, to use Aadilah's her new research on BRAF and sonic hedgehog, you can look at the interactions between these different signaling pathways. So it gives you a lot of really valuable and complementary information to, say, for example, your NGS approaches or your protein profiling approaches. So now, how can this, or how, just to give a few examples of perhaps applications or images related to our department and research that might be occurring in our department. So here we see pancreatic islet beta cells detected with insulin antibody. The red staining is beta-catenin, which is a transcription factor, which is integral to the Wnt signaling pathway. Then the next slide, I'm actually involved in a project at the moment looking at lipid accumulation in monocytes with Dr. Brett Mansfield. And so here, this is not actually our images. I struggled to download our images yesterday. But just as an example, we have isolated monocytes from patients, from hypercholesterolemic patients, and we're looking at cholesterol distribution in these monocytes. At the moment, we use centrifugation to separate these cells, but we're going to progress to a magnetic cell separation technique to specifically isolate our population of monocytes. And then to look at the cholesterol distribution using confocal microscopy. The next slide, lung cancer cell on the left, and a really beautiful type 1 pneumocyte on the right. Both confocal microscopy images. I think there's one more image. Yes. So confocal laser images of the glomerulus. So these were kidney sections that were immunostained with alpha-2C adrenal receptor and other markers of different segments of the glomerulus. And the nucleus was stained blue with the fluorescent dye called DAC. So you can see that you can obtain a lot of information using this approach. I think that might be my last slide. So just also to mention that the confocal microscopy is a well-established unit. We have an established training platform, and we can advise and help people on how to progress, how to proceed with their projects in terms of how to prepare the tissues, how to choose or what antibodies to choose, and also the secondary antibodies to choose so that you can carry out a multiple labeling approach for your work. Okay, thanks.
Thank you very much. In the interest of time, I'm going to introduce Prof. Alisha Wade. She's our second clinician research professor, and she's going to present her research interest. Thank you, Alisha.
So good morning, everybody. So unlike my predecessor or my preceding speakers, I actually started out life as a clinician. And I went into research because I was interested in what I was seeing, things that differed in the populations in which I was working, and how the outcomes in my patients were differing from what I was seeing in textbooks, for example. And I can say that although it does involve a lot of trade-offs and a lot of compromises, I have found this combination of doing research and still being an active clinician, because I still see patients, very rewarding. So if any of you all is thinking at the back of your mind whether or not you may want a career as a clinician scientist, come talk to me. I'll tell you the truth about it, but understand that it's also very rewarding. And so I've recently started a new division at the Wits Health Consortium called RHYME. I spent a lot of time coming up with this acronym, looking really at research in endocrinology and metabolism. And really my focus has been very much on health disparities in endocrine and metabolic disease, with a specific focus on populations in sub-Saharan Africa and in African ancestry populations. I think it's a reasonable question to ask, Why focus on the health disparities lens? And I think you will have heard either implicit or expressly stated through what everyone else has said this morning, that there are definite differences in what we see in our populations or our populations of African ancestry and what you see in the rest of the world. And we know that the prevalence of endocrine and metabolic diseases is increasing. I mean, if Stats SA's data are to be believed, diabetes is now the leading cause of death in women in South Africa. And we know that the approaches that we take to patients are largely adopted from data that we're getting from other places. You know, they're data from usually high-income countries and data usually from European ancestry. And we sit here Friday after Friday after Friday, and hardly an academic meeting goes by when we don't hear the phrase, but we have no local data. We have no data from our setting. And that's really important because we don't understand how disease differs, because disease does differ. We don't understand when it does. So are there particular populations or particular groups of people that we need to focus on? And we don't understand why disease differs. And a lot of what Clement and Zané and Aadilah, Raquel and Alisha has said has been really trying to, in many ways, get at that why disease differs. But a lot of that has been from a molecular perspective. And my approach tends to include the molecular, but also expand that a little bit more. So I have particular areas of focus, which is unsurprising as an endocrinologist, they're endocrinologic. So diabetes, thyroid disease, both from a structural and a functional point of view, female reproductive health, and also looking more and more at non-communicable disease multimorbidity, where we have more than one non-communicable disease occurring at the same time. But I try to take, again, as I'm not a bench scientist, so I don't have all of these wonderful technical skills or toys and pretty pictures like Clem. I take a very broad approach. And if you'll remember the diagram that Raquel showed, I'm towards the kind of the left of that circle. So my research approaches deal with the epidemiology. So who's presenting with disease and why? Because I think that whenever one is asking these questions, it's really important to have that foundation. Before you want to spend a lot of time, effort, and frankly, money investigating these things, it's important to define how do we differ? So what's our competitive advantage? Why are we looking at this in a South African, Sub-Saharan African, African ancestry population, other than it hasn't been done before? What do we think that we're going to find that's different, that's going to be able to move the science forward more broadly? So I use a lot of epidemiologic approaches. I also use some of the approaches in collaboration with my molecular biology colleagues looking at mechanisms. So we're doing some really exciting work now looking at thyroid functional disease in African ancestry populations. And what you see, if you care to look, is that if one looks at the distribution of thyroid stimulating hormone in African ancestry populations, it actually has a different distribution than European ancestry populations. And we're doing genomic research now to try to understand why. You know, are there actually differences in which genes inform how the thyroid pituitary axis is controlled in African ancestry versus non-African ancestry populations, and finding out really interesting information. And in addition to that, also really important is health outcomes. We can't divorce the fact that we work in a constrained healthcare system, and how much of the outcomes, or what are the outcomes that we see in our patients, are purely driven by the molecular biology. What of it is driven by that interaction between the molecular biology and the environment, and what of it is driven by our health services, such as they are. And in terms of when one considers that our overarching goal, or the goal at the end of the day, is to improve patient health, do we also need, we know we need to do interventions also in terms of health services research, implementation. It's great to come up with all of these wonderful things. You know, Ismail spoke about precision medicine, Zané referred to it as well, but at the end of the day, these things, we have to be able to work out how can we actually implement these in a clinical setting, and how can we actually translate these findings and make them accessible to patients if we actually expect to have an impact and improve our patient outcomes at the end of the day. So part of what I'm really interested in is also how do we foster skills transfer? So how do we take all of this, all what do we know, and how do we move it into the next generation? And as a result of that, I'm very keen on always talking to young, or maybe not so young, enthusiastic people who want to transition into research. And some of the skills that I can offer, help that I can offer, things that people may be interested in and not know, well, how do I get started? If somebody wants to write a grant, I mean, we can't write them for you because we're busy writing our own, but always happy to read, to comment, to give feedback. Who can I talk to? How do I start? Where do I even start to look? Those are some of the things that we're always keen to help. nested master's and doctoral projects. So again, people have spoken about that. We always need more help and more hands, but working within an established project is always a great way to start picking up skills. And many of us have projects which are, kind of nicely distinguished and delineated that are just waiting for enthusiastic people who want to use those projects as master's research or doctoral research. Right? Even if you don't want to do a degree, though, Ismail, cover your ears, even if you don't want to do a degree, because the university wants us to do degrees, which is great, but sometimes that's not where your life is taking you, or that's not where your career is taking you. You don't want to do a PhD, but you want to do research. And there are lots of avenues to do research that don't necessarily mean that you have to register for a degree. Come talk to us, because we have lots of those opportunities available. And then lastly, I think there are lots of opportunities for other skills development. Article peer review, for example. Many of us have more requests for article peer review than we can possibly handle. If those are things that you're interested in doing, let us know. And the idea is not to kind of throw you an article and we'll help you through it. We'll talk to you about it. We'll say, we'll sit down so you can come up with a framework as to how do you approach reviewing an article. And those are some of the things that we could do. I think as time goes on, we'll discuss how we look at the research unit and how we can kind of develop frameworks for interacting so people know what's going on, know what the opportunities are, and know how best to take advantage of them. Thank you very much.
Thank you very much. Okay. Are there any questions from the platform? Anything online? Oh, Dwayne. Hi, Dwayne. Come through. Sorry, my apologies. So, do you have a slide deck? Okay, all right. So, hi. Sorry. Okay.
Okay. Yeah, so I'm the research coordinator for the school at the school level for the cluster, which includes internal medicine. And what I do mainly is to provide support for NMEDs. So in terms of you're thinking of your research question, you're still not totally clear, I can assist with that. You're thinking of how do I design my study? What can I do? Objectives, thinking of data analysis, some guidance around that, different levels of support in terms of the NMEDs, and also some of the things that Alisha talked about, mentored peer reviews and things like that, guidance with writing. And also, I'm sure you've seen a few emails from me, at least the NMEDs must have seen a few emails from me with information from the faculty. So basically anything that has to do with your research and research questions, how to handle it. Yeah, that's more or less what I'll do.
Thank you. Thank you very much for that, Dwayne. Just to reiterate, so Dwayne is the research clinician scientist in the internal medicine cluster. So she offers a significant amount of support in terms of, again, data analysis, statistical analysis. She sits on the WITS HREC, so if you want any advice about how to develop your ethics proposal, she's there to assist. Quite a few of us, there's at least about 4 of us who sit on the ethics committee in the department: Nina, Diana, Sarah Stacy, Raquel, Duarte. So please, if anyone's having a problem with ethics, again, please contact them. They'll be more than willing to assist with the ethics. So all of these terminologies and these presentations might seem a bit overwhelming, but they really aren't, right? They actually what's happening, and we're actually lagging behind the curve if we look at what's happening internationally. In terms of our undergraduate educational platform, because our students are not being exposed to these sort of topics. I know that the students complain a significant amount about molecular medicine, MolMed. I know my daughters had a tough time with MolMed, but this is all basically what's happening, right? So MolMed, this is MolMed. This entire presentation was a whirlwind tour of molecular medicine, and it's from its origins right up to the expression of the disease entity on the clinical platform. So just to get back, are there any questions? Anyone who wants to engage with any of our clinician scientists about any specific projects that they have in mind? Jared, anything online?
Nothing online.
Nothing online. All right, so we're almost at the end. I think we're at 9 o'clock. Thank you very much. It's a very well attended meeting. I'm really appreciative of your presence. And yeah.
Everyone who's presented today, are you happy for me to send out your contact details to the mailing list?
Yeah, and just include Marilis and Busi on the email on them. So
So then if you don't want to ask in the room, you can,
Yeah.
you can send an email or phone.
all right. So please, it's a phenomenal opportunity. It's a bi-directional opportunity. We're looking for students to assist us on the clinical platform, on the research platform at the bench side. And we're wanting ideas to come from our clinicians. So senior all the way right down from junior. If you don't bring and ask research questions, our clinician scientists will never be able to know how to answer those questions. And they're looking for opportunities to translate the work that they're doing in the division just behind us, in the area just behind us, back to the clinical platform, right? So we're really trying to establish that link and try and promote it so that we become one of the forerunners in research. And it's an important aspect for our alumni. So all of you are WITS alumni, most of you, at least I hope the majority of you are WITS alumni. And we want to be the leading research university in South Africa and then in the country. And in order for us to do that, we need to have a research ethos and a research focus. And this is one way to develop that, to develop this conveyor belt of research in which students can just plug into and come out with research papers, come out with research degrees, and hopefully foster a research project that will be sustainable in the department. Okay, thank you very much. Thank you, Jared, for facilitating this meeting.